Hi, good afternoon. Thank you for coming. This is going to be a bit of a change of gears. I want to tell you about my company. So it's going to be a mix between a sort of a position statement and a product pitch, really. As mentioned, the background to this company is more than 10 years that I spent in academia working at the European Bioinformatics Institute, where I started as a programmer, and when I left it, I was running my functional genomics team, building large-scale services for the scientific community. And what I encountered was that in order for us to build the various tools, and one of the tools, for example, my team built was a large expression data repository with various queries and interfaces. In order for us to build these tools, we needed a lot of infrastructure, and we ended up inventing a lot of that infrastructure. And that infrastructure could only exist in a place like the EBI. So what I really wanted to create with Genestack was a means to provide this kind of infrastructure to just about anyone. So first, some possibly controversial statements. I would say that bioinformatics is evolving. I mean, I would come talk to me afterwards. I can tell you that it's not just bioinformatics. In fact, I think the whole software and data analytics industry is evolving rapidly. It's not about data generation anymore. It's really about data management. I would argue it's not even about data processing anymore. These days you're spoiled for choice of how to run your pipelines and where to run your pipelines, and reproducible pipelines are in every flavor, from Dockerized containers, common workflow language. You've got DNA nexus, etc., etc. So I would say that the data generation and data processing is, by and large, a solved problem. It's cheap and getting cheaper. You have a lot of choice. I would argue then that the role of a sort of standalone bioinformatician is also on its way out. It's disappearing. That's not to say that bioinformaticians are unnecessary. It's just that they should become invisible. As invisible as when you launch iTunes and you start playing a track. You don't know where that music is. You don't know if it's on your computer. You don't know how it's getting decoded. These days, still, the guts of bioinformatics are very much exposed. We expect biologists to learn Python. We expect people to, be able to write R shiny apps. I think that kind of stuff is going to become more and more invisible. As proof of the fact that this is already happening, for those of you who have been in the field for a while, consider how much effort it would have taken you to build a neural network to do the kind of machine learning that the previous speaker talked about, let's say 15 years ago. Yeah, it required pretty specialized knowledge, quite a lot of programming. To do some deep learning AI about 4 or 5 years ago, you could go to the emerging technologies like TensorFlow, and still it requires a fair amount of investment. These days, you can write it literally in 5 lines of Python code using Keras. So this kind of complex software is becoming simpler and simpler and easier to write. So what remains difficult? Well, we're getting more data, and we still need to analyze it. And in fact, getting a grasp of data and the analytics is what's difficult. And if you look at what people do today with data, they generate it, they load it onto a cloud, onto a local cluster, they analyze it, they produce a report of some sort that ends up in a PDF and a PowerPoint, and then it dies. So what we want to do is really think about how can we extend the data lifespan? How can we make it live longer? So there is a tremendous amount of data being generated. If, let's say, looking at data sets that were generated the last, say, before 5 years ago, we were talking about thousands of samples, that's sort of the 1,000 genomes, TCGA. There are initiatives to aggregate data, and we're talking about tens of thousands. So Nomad was released a month or so ago, and that's got about 16,000 whole genomes reprocessed, reanalyzed. There are national genomics projects that are pushing the data sizes into hundreds of thousands. And then, people with very deep pockets like AstraZeneca, HLI, they're talking about millions. So we need to build ecosystems to make it possible to work and manage data of these sizes. So what are we doing about it at Genestack? We're trying to create an ecosystem that's going to bring together data, tools, and knowledge. And by this, I mean the following. We're trying to build a system where you can organize, search, collect, and aggregate data across different sources. So data from public domain, data that is produced internally within an organization, data that is shared with collaborators. So to be able to search across various metadata attributes, put data together for meta-analysis, while at the same time imposing strict access controls so that one can control who has access to what data with what permissions, keeping track of, if necessary, consent, things like that. Next, we want to be able to, as I said, get away from the idea of very visibly exposed tools and pipelines. And to do that, you need to have a very flexible deployment model. If necessary, we want to be able to run on the cloud, run on Amazon, or if need be, and use Amazon virtual machines, use Dockerized instances, use containerization. Or, if necessary, we want to be able to run on an existing HPC environment and plug into a local distributed data storage system, use your LSF, what have you. And then we want to be able to do analytics interactively, so that you don't have to wait, to do a variant filtering. You can do clustering on the fly, so you can do interactive visual analytics at population scale. And I'm curious to know what it's going to take to get there. So let me show you some of the things that we have done at Genestack to make this possible. So one of the things that we do is we've invested a lot of effort at managing data and metadata. And the whole system is accessible via the web or via a scripting interface. And we provide a really powerful, flexible mechanism for describing data, ranging from describing large projects, experiments, studies, all the way down to individual data types. And the way that our system is built is that different data types have their own descriptors. So if you're talking about, let's say, a sequencing assay, you can decide what attributes it has, from which public or private ontologies these attributes must come from. If you're talking about arrays, or if you're talking about VCF data, all of these data types can be described and annotated. These templates control how data is described. Templates can be shared with partners and collaborators, thereby making sure that if there is a data producer creating data, then these data will be described how you want them. If you're working in a large enterprise context where you have a LIMS system, we can integrate with the LIMS to populate it or to retrieve data from it. Once you have these templates set up, then data can be imported, and then data control validation reports will be produced, so ensuring that data is indeed described the way that you expect it to. And we've developed lots of tools to help people create data sensibly. So this means that when you want to import data into Genestack, the system recognizes automatically what kind of data you look at, tries to extract metadata from the files. When you are entering data, then if there is a complex key with a dose, a unit, a time point, then we automatically provide various autocompletes and so on. And having built this interface for data ingestion means that we can do some pretty neat things for data use and consumption. So we can build pretty powerful search interfaces. So you can now search across all the metadata attributes. If you're looking at numeric data with units, you can do unit conversions on the fly. So if you've imported data and was annotated with micromolar concentration of a chemical compound, then you can search for nanomolar, or you can search for molar concentrations. We'll do the unit conversion on the fly and find it. We have used this mechanism to load into our system pretty much all the public data that we could get our hands on. And this is a constantly going process. So when you use Genestack, you have access to all the data from public repositories. So we're talking about Geo, Array Express, ENCODE data, Ensemble reference genomes, you name it. All that data goes through this pipeline. It gets mapped to existing ontologies. It becomes findable, searchable. But what's more is once you find this data, you can right away start to view analyses. If an analysis already exists in the system, then from the data search interface, you'll see a link to go and view this analysis. If it doesn't exist, you can start one. And because we have all the metadata available, we can make suggestions as to what sensible analyses might be. Yeah? The whole system is built as an operating system might be. And so it means it's not a closed system. You can add tools to it, you can add applications, you can add your own tools, and you can add your own analysis types. So everything that you see here, all of these screens are written using our SDK, which is made available. So all of this can be integrated and extended. So it means that right away, you can go in, you can do a search across data that's from public domain, from private, share it with you. You can create large-scale cohorts, aggregate them, and start doing analyses. What kind of analysis can you do? This is just to run through some examples of the kind of things we have done. So we've enabled our system to understand chemical structures so that when you annotate data sets, you can describe with which chemical compound, the drug, something might have been treated. And we understand the structure underneath, so this rendering of the structure is in fact in real time. And this means that we can do searches such as, give me all experiments where my tissue was treated with a compound that has a structural similarity to this one. Yeah? We've reanalyzed a lot of public drug response data and computed the IC50 curves, we computed all the fits, and now we can do pretty interesting meta-analysis. So we can look for, give me all chemical compounds that are structurally similar and have a similar gene signature responding to treatment, or look for common IC50 values. So we can do this kind of analysis. And all of these tools, again, are composed using our SDK, using our components which are available to use. We also have been able to integrate genomic data. And again, the idea is to allow you to access large scale of genomic data types, aggregate them and run them through a pipeline without having to get into the coding too much. And it gives you visual interfaces for doing population stratification, doing association analysis, and being able to drill down all the way to individual genotypes. So this is work we did with a hospital in Manchester. Initially started looking at exomes and moving on to whole genomes. There are tools to do visual QC and filtering from raw reads QC all the way through to aligned and then variant association mining. There is an interface that allows you to interactively query variants across thousands of individuals and millions of variants. So, and this is on the fly, and all of this can be scripted from outside the system or can be accessed internally. Once you have identified, and this works for every app in the system, a final result, you can lock it and share it with colleagues. Another neat feature that we have in the platform is that for every result, there is a mechanism to obtain the precise provenance of how this result was computed. And this is what we call a reproducible data flow. There's a picture here at the bottom of the slide. What does it mean? It means that for every result that's computed and created in the system, we record the metadata and we record the dependencies of this result on all the inputs, all the tools that were used to create it, all the parameters, all the versions, and we can then reproduce the complete graph of dependencies and display it. Then you can also export this graph as text for your methods section of the paper, but what's really neat is that you can take this graph and you can say, well, this is my pipeline. I want to be able to reuse it. You can say that, okay, now I want to take one of the nodes in this pipeline and then put in different data into it. So you could do something as an experiment on a couple of samples. Once you've got your pipeline working, you can then reverse engineer the result and rerun it on hundreds or thousands of samples easily. So where does this then lead us? It leads us to the ability of doing on-the-fly, fast cohort selection and construction using available data, results, and metadata. We can then run these through scalable pipelines with optimized algorithms, and we have ability to deploy this and externalize the pipelines if necessary. So we have our own mechanism for running pipelines, but we don't care which pipelines we use. We can use, we've integrated our system with open source pipeline engines or commercial engines. And then we have a fast storage and query engine for interactive visual filtering of data. So this is data and tools. Where do we go from here? Well, we want to go to some higher level knowledge. And this is work that we've been doing with Rothamsted Research, which is a research institute in England. They do a lot of agricultural research, and they've developed a number of tools for building gene trait networks. So these are tools that combine very diverse data types such as GWAS, traits, publication data, protein-protein interaction data. They create, you define your different input sources, you create a network that integrates across these concepts, and then there is a visual exploration and data mining tool for exploring these networks. Now, these tools are really, really good, but they've only developed them for a number of organisms in the plant world, and these tools are updated maybe once every 6 months. So what we did is we migrated these tools to our platform. We hooked them up to all the pipelines and all the data processing steps that we already have, and we made it possible to create these networks in a matter of a couple hours. So what does this now mean? This means that within Genestack we have public networks that integrate very, very different data types for plants, but we also have now the ability to extend these with private networks. So you can now take, again, public data, you can add to it private data, you can run it through a pipeline, and then you can start building and sharing networks with colleagues and incorporate information like homology, expression, reference networks. The system provides a visualization, exploration tools, and really makes it possible to go from finding raw data in public domain or from collaborators, doing the primary and secondary analysis, and then really moving it into the land of tertiary analysis. So I have a couple of words about how this thing works. So it's a very modular, scalable OS-inspired design. There are really sort of three main components, and that is the apps and the virtual file system that we introduce. And that's what you generally tend to interact with. And then at the back end of the system, there is two abstractions, an abstraction for storage and an abstraction for compute. And what that means is that our system can be deployed pretty much anywhere. You can run this system on the cloud, and we have a community edition on Amazon. It can be run on premises, integrating with existing infrastructure, or as a hybrid model. We have a mechanism for reverse engineering results, so thereby producing data flow and file provenance reports. And the system includes ability to do search and metadata control using ontologies. So before I finish my presentation, a couple of thoughts that I wanted to throw at you so that the software engineers and bioinformaticians in the audience don't feel like their time is completely up. There are some interesting challenges that are still ahead of us. This is a sort of a mental exercise as to what are really the upper bound for a system like I'm describing that are supposed to work at true population scale. Just sort of to outline where we want to go. So consider this. If we wanted to capture diverse genomic data, yeah, and if we wanted to capture it at a very granular level, let's just say that we're going to take one data point per nucleotide. So this might be variation, or it might be methylation levels, or it might be transcription at allele-specific expression. Let's say that we're looking at also a number of phenotype measurements, let's say 1,000, and we're looking at a world's population. Now we want to do this at every single cell over, let's say, a lifespan of 100 years. So this is quite a lot of data, yeah? And this data is generated in real time. we're talking about allowing for 4 bytes per point. Yeah. Now this is an approximation because some of this data is going to be discrete, a lot of this is going to be sparse, but quite a number of it is going to be floating point. So let's say 4 bytes per point. We're looking at 10 to the 26 petabytes. So this is quite a few orders of magnitude of what we can work with today. Yeah. So the challenge for a true population scale genomic system is quite beefy and it's still out there. Yeah. And you want to be able to add data to this very large cube in real time. Yeah. So as data is generated, and you want to be able to query, compute, and do summaries on sub data cubes within this. Well, maybe not everything is not so bad. So this is what's going to happen probably quite a few years from now. Well, what are we going to be doing this tomorrow? So if you look at tomorrow, it's not so bad. We're looking at maybe 100,000 genomic data points per individual. Let's say we still have our 1,000 phenotype data points. We're looking at maybe 10 to the 7th in terms of individuals. That's the data size that we're looking at. We're not yet able to survey all the cells in the body, maybe 1 million cells, and looking at maybe 1,000 time points. And this now becomes a lot more manageable, right? 4 million petabytes. And I think that those data sizes are not that outlandish. So that's what's happening tomorrow. And as for today we're pretty much able to work with data sizes that you can almost fit into memory. Yeah? So today, we're looking at maybe, again, 10 to the 5th data points on the genomic data dimension, maybe 1 million individuals, yeah, or a few million. We're looking at only a few cell types at a time, and maybe only 10 time points. And so this is a data size that's now in the tens of terabytes range, and the way that the computer systems are evolving, I put it to you that it is possible to build systems that allow for fast, interactive, scalable analytics on this kind of data, and that's what we're working on at Genestack. So up until recently, we've been working mostly with very large pharma companies or consumer product goods or large universities. We're very keen to get our hands on data to really push our system to the limit. So if you have a large omics data collection with very diverse, interesting R&D usage requirements, we want to partner, we want to collaborate with you. So come talk to us. Thank you.